Okay, we are now recording. So I want to thank you all for being here. I'm pretty excited about this. My name is Matt Van Hook. I'm here at the University of Nebraska Medical Center in our Department of Ophthalmology in the Trolson Eye Institute. Um, we have what I think will be a really informative topic for this virtual discussion today. This is one that I am especially interested in, as you know, I'm a uh, uh, fairly basic level scientist. I do laboratory work on mice, and I have very little insight into the ins and outs of the regulatory process and how clinical trials actually work. And I know within the RReSTORe consortium, we have a wide breadth of people with varying levels of experience, including many of us who are exclusively labs, some of us who are clinicians, and kind of everything in between. So, the goal of this is really to give all of us an exposure to the regulatory process, because if our goal is to be able to advance therapies for vision restoration, that will eventually require clinical trials to make that happen. So that's moving stuff from the bench all the way up to the bedside and all of the steps in between. So we have a wide array of experts who can share from their experiences with this process, with the group. And again, the goal of this is to really have it be a discussion. So each of them has prepared about a 5 to 10 minute presentation, kind of sharing some of their experiences and relevant background. The goal of that is to be able to spur discussion. So they'll have some time to present. And then we're going to open it up for the remainder of our time for everyone who's in attendance. Feel free to chime in by unmuting yourself and asking questions that way. If you want to raise your hand virtually during the discussion, feel free to do that, and I'll help kind of facilitate that and make sure people get a chance to speak. If you'd rather, you can also put questions into the chat, and I'll do my best to make sure that those get directed and repeated and answered by the group. If you have experience on some of the topics that are coming up, you're welcome to chime in with your experience as well. This isn't just for our discussion facilitators to field questions. This is a discussion that's opened up to everyone in the group, so it's broadly open to us all. So what I would like to do before we start is I just want our facilitators to briefly go around and introduce themselves, tell us who you are, kind of a little bit about your experience and your institution affiliation. So I'm going to start with Barb. How about you say hello?
Okay, great. Thanks, Matt. So, hi, everyone. Barbara Wirostko. I am a clinician scientist. Been in large pharma, small pharma, have started a few companies and I am not a regulatory expert, but I've done several INDs, clinical trials, both at Pfizer and now with some private companies. So I wanted to give you guys a background on some of the work that needs to go into that first IND submission.
Wonderful. Gee,
Hi there, my name is Chi Cui. I'm an associate professor, University of Pennsylvania. I'm also a clinician researcher. I would describe myself as more as of a consumer of the policies regarding regulatory processes as opposed to an expert, mostly because I'm sort of involved in the process of translating from the bench to the bedside right now. So I've had to do a lot of homework and hoping to share a little bit of what I learned during the symposium.
wonderful. Thank you. Gus.
Hi, I'm Gustavo Moraes. I'm an associate professor at Columbia University, New York. And my interest has always been on glaucoma treatment, particularly endpoints and how to improve our understanding of endpoints for clinical trials. More recently, thanks to work I do at a clinical research organization, I've been able to interact with the FDA quite often on these aspects, and I'd be happy to share some of that insight here today.
Wonderful. And Aki.
Hi, my name is Aki Shukla. I'm a glaucoma specialist and associate professor at Columbia University. Excited to meet with all of you today. I recently went through the IND process for a clinical trial that is ongoing here at Columbia, looking at the role of nicotinamide in neuroprotection for glaucoma. A lot of this was new to me during the process, and I still feel like I have a lot to learn from the folks on this call, but I'll share with you the experience that I had.
All right. So our first presenter is going to be Barb. She will now share her slides and take it away.
Okay, great. Can you see that?
Yes.
Everybody can see my slide? Perfect. So in my role, I still have an academic appointment. I'm at the University of Utah, which is great. We really have a fantastic glaucoma department. And I did fail to say that I'm a glaucoma specialist. So again, topic is near and dear to my heart. Really no disclosures that are relevant to this talk, but what I wanted to call out, and I think is such a great resource, is CIRM. CIRM is interested in funding everything from early discovery through translational, even through IND into first-in-human phase ones. And it's all on stem cell and gene therapies, so regenerative medicine. So again, and a lot of the work they do is support bench scientists who have an idea or starting a company, or even the company stays within the university. Again, just a phenomenal wealth of knowledge, access. I've been a subject matter expert for them for years, and I really invite you to check them out, because if you do have some foothold in California, they can support you with their grants. And it's non-dilutive funding, which means they don't take any part of your company. So again, just a great wealth of knowledge. So when we think about the preclinical work, and this is really, really important, is what the regulatory agencies are really focused on is safety. They want a concept of efficacy. They want to know that what you are doing makes sense from a biologic perspective, but really the whole remit for an IND or IND-enabling tox is to ensure that your stem cells are safe and effective for humans. They're going to ask for biologic plausibility. They want to identify the doses and assess safety and feasibility, again, for that first-in-human study. So they want to know, how long are you treating? What's the maximum dose? And when I think about the amount of work that you will do preclinically, it's really to validate or mirror the clinical exposure. Now, that's really easy with a small molecule, because if your small molecule is going to work, for 20, for a day, 24 hours, 48 hours, and you want to do a 1-month study in humans, then you give them 1 month of data preclinically. It's a little bit more complicated, obviously, when you get to stem cells and gene therapies and even biologics. So the importance of the data, it's vital to support, again, that transition from preclinical research to clinical trials. And again, focuses on safety. So they want to know that the way the clinical trials are going to be conducted is there are appropriate safety considerations. So what is that? I always think about it as thinking about what that first-in-human study is gonna look like. What's, the duration? What's the design? What's the endpoint? So your foundation for that first-in-human study is really set up by your preclinical work, even as you're thinking about biomarkers and endpoints. If you're going to be looking something simple,
I don't know whether
IOP,
even
right?
if they take it, why he didn't take his own one? Because he said he
Thank you. So you want to use, again, the same endpoints. The other thing that's really nice too is you can look and explore novel endpoints in those preclinical studies as well, especially if you're working with a diseased model. Toxicology, they're going to be concerned about the local tolerability and tox profile, but also systemic. So again, with small molecules, if you're delivering them topically, you're going to have to deliver them at higher doses systemically. You're going to try to tease out what's the worst safety concern that could occur. The division that handles the biologics is the CBER. So CBER, which is different than CDER, which is for drugs, and also different from devices, which is CDRH. This is the division. Again, they've got great guidelines, and specifically under the biologics division is OCTGT, which is the Office of Cellular, Tissue, and Gene therapies. And again, I'm going to run through a lot of this quickly, and but I can provide these slides so that, again, it's a little bit of a background. As the FDA website says, there's no specific guidance on all the preclinical models, but there are guidance documents available, webinars. And I think the key thing here is the FDA wants to work with you. And I know, Aki and Gus are going to talk about this as well, but they want to get therapies into patients' hands. So again, you can work with them early. They are open to discussions and really being creative on endpoints as well as the models. So here's some example of stem cells. Obviously, everybody on the call is familiar with this, but they think about the adult stem cells, perinatal, so the stem cell-derived cells, as well as functionally mature differentiated cells. And here's just some of the safety considerations that they start to think about. And this is actually off of one of their slide decks, is they think about cell migration. Where are the cells going? Are they going to non-target sites? And what's going to happen when the cells get there? Do they differentiate? Do they actually mutate? What about ex vivo manipulation? So what's happening to those cells when you're growing them? What are the processes and the procedures and the analytical work that has to be done to make sure that those cells are the same cells from beginning to end as you are, I guess, expanding on them, right? Or cloning them or growing them. What are the potential inflammatory immune responses, right, with these cells? Can you get inappropriate cell proliferation, so tumors? Can you get ectopic tissue formation? And then one thing that really comes up again and again and again is, and it's back to safety, is what's the immunosuppressive agents that are being used and in what patients. So one of the things that we discuss a lot at the level of the CIRM grant reviewing committees is those patients who are vulnerable and they're now perhaps immunocompromised, or we're immunocompromising them with the agents that we're giving. So again, it goes back to safety. Can you make a good argument for why this product should be put into a patient based on the unmet need? The other key thing to think about here is the device or the mode of delivery. So if you are delivering a stem cell via a unique injecting system, that is now a separate device that needs to go through CDRH. And you're going to need to get that device cleared either in combination with the product and or standalone. And they also want to know, again, safety. If you're injecting those stem cells, what is the device injector doing from a safety perspective as well? So it can get complicated, but if you keep coming back to safety, safety, safety. Again, some more of their considerations. Is there a scientific basis for even conducting the clinical trial? What's the feasibility? What's the rationale? Do you have proof of concept? So again, thinking about those preclinical models in mice, or I love what Luxturna did with retinitis pigmentosa. They were able to use a diseased model to show basically development safety. They used it for their IND, and they were able to work with the FDA on a novel endpoints for visual function under different conditions of illumination and things like that. So again, the FDA can really be your partner. They also want to know your dosing regimen and your methods of dosing, obviously. So the risk benefit, we talked about. How are you going to study your compound or the stem cell or the product in your first patients? Is it a single ascending or a multiple ascending dose study? Again, what's the target tissue? Are there off-target toxicity issues? And how are you going to monitor those patients? They're very concerned about the monitoring. You can do a safety monitoring group, data monitoring group, and also what's the eligible patient population. Obviously much different if it's an older patient with end-stage disease versus younger patients or healthy individuals. Dose exploration, proof of concept in your initial studies leading up to that IND, which is an investigational new drug application. You're going to have to show safety data up to certain limits. Again, you're pushing on safety. What's the highest amount of product you can give before you see a safety signal? And what is that safety signal? And needing to think about it too, when we're studying a certain compound and injecting it into the eyes of healthy rabbits, their vitreous is very different than the vitreous of an elderly patient who has diabetes, that may have had a vitrectomy, that may have, vitreous syneresis. I know you guys have discussed it at some of the RReSTORe meetings, but the difference in the internal limiting membrane, how is that going to impact cell migration and efficacy, right, in different ages of animals as well as different ages of patients, and even the size of the patient, right? Could that also be a difference in efficacy and safety? Key considerations, you're going to hear terms like GLP, good laboratory practices. That's what you need for the IND tox work. These are compliant toxicology centers, generally contract research organizations that do this, professionally. There's good manufacturing practices for your product, and then there's good clinical practices for the clinical studies. And they follow ICH guidelines. Here's some resources. Again, a lot of great material on the FDA website. This handbook I thought was really cool. It's a little old now, but a key piece was actually the quote, "The FDA recognizes the need for flexibility in its recommendations for stem cell therapies and will consider many factors, including the target population, the seriousness of the disease, and the potential risk and benefits." So again, it always comes back to that risk-benefit profile. Why am I not moving? There we go. So in summary, preclinical studies for stem cell therapies are a critical step as we look to develop these new therapies. The preclinical work is going to ensure that the clinical trials are conducted, again, safely, and this is the FDA's words, safely, responsibly, and ethically, ultimately leading to safer and more effective therapies. And then there's really a great opportunity to engage with the FDA early through certain avenues such as breakthrough, also orphan disease, and you could do what's called a pre-IND. So again, get them involved early and start discussing. And this stem cells and, for an optic nerve regenerative therapy is a high risk, high reward and is complex. So thank you for inviting me. I hope this was helpful.
Wonderful. Thank you. Let's turn it over to you.
Let me start sharing here. Can everyone see that?
Yes.
Okay, great. Okay, so, Barb already gave such a great overview of the process. And so there's going to be a little bit of overlap with what I'm going to be talking about, what she already spoke about. But hopefully it'll help to sort of reinforce some of the key concepts here. So my focus is going to be sort of talking about trial design and sort of the process for gene therapy. And I'm going to specifically talk about Luxturna as a case study for this whole process. So trial design for gene therapy really benefits from a lot of regulatory documents that are available. And I'm going to share all of these references with Matt, and Matt, you can share with a group afterwards. So as Barb already said, the goal, of course, is to balance safety, efficacy, and regulatory compliance. So both the U.S. Food and Drug Administration and the European Medicines Agency, which is the European counterpart to the FDA, have provided extensive guidance documents on gene therapy product development, preclinical testing, and clinical trial design. So most of what I have in this talk pulls directly from those documents. Specifically, the FDA has a guidance document called Human Gene Therapy for Retinal Disorders. So exactly what we're looking for if you're thinking gene therapy, or sort of some of the details they talk about actually applies to other biologics for injection into the eye as well. So there are 4 objectives for a preclinical program, one of which is to identify dosing level and range, dose escalation schedule, and dosing regimen. Number 2 is to establish feasibility and safety of route administration. Number 3 is to provide support for patient eligibility criteria. And number 4 is to identify toxicities and physiologic parameters that guide clinical monitoring. And specifically, the use of non-human primates is encouraged and, in fact, required. So what are some key considerations in trial design? So number 1, it's really important to understand the natural history of the disease, right? Without a clear understanding of how the disease progresses, we don't really know when to intervene to show efficacy in our therapy if it's available. In terms of study design, it's recommended that we use randomized concurrent parallel control groups, masked whenever possible for obvious reasons. And because we have 2 eyes, it is recommended not to use the contralateral eye as control. In terms of the study population, we want to confirm the targeted genetic mutation or the disease is in fact present. In terms of benefits greater than risks, as Barb already talked about, first-in-human gene therapy trials should enroll patients with severities of visual impairment that offer favorable benefit-risk profiles. So that's a little bit open for interpretation, but certainly, our goal is to first do no harm, right? We want to help these patients. We certainly don't want to make them worse. It is important to include children as appropriate, specifically for retinal degenerative processes. A lot of them affect children, but often it's recommended that adults should be tested first. With respect to implementation, the purpose of an early phase trial is to compare a range of doses to assess safety and efficacy, treat the more advanced eye first in bilateral disease. This is something that was in one of the guidance documents. And in terms of study protocol, there should be a detailed administrative plan, including delivery procedure, delivery devices, and dosing schedule. With respect to safety considerations, it is important because some of these delivery of these gene therapies require, intravitreal injections or subretinal injections, utilizing trained individuals to minimize the risks associated with intraocular delivery is very important. The consideration of local and systemic immune responses is certainly important as well. So I'm not going to belabor this point because I think Gus and maybe Aki are going to be talking about it. And, Gus is certainly an expert in this point. But endpoint selection and measurement tools is very important, right? So early phase is focused on efficacy. So ideally, there will be functional and structural endpoints. And late phase must provide evidence of effectiveness. Specifically, here are a few points that are outlined in these documents. Best corrected visual acuity improvement. So this is halving of the visual angle represented by a gain of at least 15 letters on the ETDRS chart. And change in a rate of photoreceptor loss. And this is measurements between the baseline and at least 2 subsequent images with an interval greater or equal to 6 months. And also a visual field change means that a mean change greater 7 decibels and greater than 5 pre-specified points is considered meaningful. Some of these endpoints, as we think about it, can be a little bit difficult to meet. And so in terms of sort of coming up with endpoint selection and measurement tools, the FDA does encourage sponsors to develop and propose novel endpoints to measure clinically meaningful effects for surrogate endpoints that are reasonably likely to predict clinical benefits. So here is where an understanding of the natural history of the disease is really important. If we understand how the disease is affecting the eye, then we can propose meaningful endpoints that may not be these pre-specified endpoints written in these documents, but nevertheless can be accepted by the FDA. And I'll talk a little bit more about that, talking about Luxturna as a case study. So I won't talk about this next point because I think Gus will probably talk about it. And I'm going to skip ahead and not talk about sort of the approval process because I think Aki is going to talk about it a little bit, so I won't belabor that point. And I did want to mention this regulatory approval and compliance regulatory playbook. So this playbook is available online, and it really talks about the different processes that we need to get from preclinical to actual clinical approval. And it sort of outlines the process in a fairly linear way. So anyone who has an interest in sort of figuring out, what's next after this step and what is sort of the end point I'm going to get to can look at this playbook, and it can be helpful. Okay, so let's talk about Luxturna. I mean, we all know what that is. It's the first approved ocular gene therapy, first by the FDA in 2017, by the EMA in 2018, for treatment of RPE65-mediated inherited retinal disease. This is a gene that is a package in the AAV2 vector for delivery via subretinal injections. One thing that Barb already alluded to is something that Jean Bennett and Al McGuire sort of had a really ingenious way of figuring out endpoints for their particular studies is they developed something called multi-luminance mobility test. So the idea here is that a lot of these patients, by the time they get to the point where they're a candidate for gene therapy, already has very, very advanced vision loss. So if we're trying to figure out whether these patients are improving after receiving gene therapy, they needed to come up with some new way of testing this. So they basically came up with an obstacle course where they can sort of edit based on patient factors, based on where they are in the trial process. And they were able to get this approved as one of the endpoints through a collaborative process in talking with the FDA. In the latest follow-up, which was published in 2021, there's long-term functional improvement that has persisted at 3 to 4 years follow-up. So if you have any interest in this process, I highly recommend this article that was written by Jean Bennett and Al McGuire and published in Cold Spring Harbor Perspective in 2023, where they really document the process that they went through and all the trial and error and all the lessons that they learned, bringing Luxturna to market. And I'll highlight a few of these. Number one is the route of administration. They looked at different types of route of administration. What they realized was that intravitreal resulted in inner retina and visual pathway transduction, but it also increased the inflammation in the eye. Eventually, they ended up doing subretinal injection because that resulted in outer retinal transduction, which is the area that they're looking for. Very little inflammation, and was also unaffected by prior AAV inoculation. When extrapolating from animal models to humans, and this is always a difficult process, with respect to gene therapy, what they figured out was this calculation of multiplicity of infection, which is calculated based on the number of cell types of interest in the treated area, is more useful than total dose. And this makes intuitive sense. In terms of delivery device, initially they were losing a lot of AAVs with every injection, but what they realized was that these AAVs were actually sticking to the inner lumen of the needle as they're doing the injection. So when they added a non-ionic surfactant, this increased their delivery efficacy. So this is also a trial and error process, not something we necessarily always think about. They also highlight the power of social media. Now, this is something, since their trial was conducted, we have a better understanding of, certainly, but, trial recruitment is always difficult, especially for a rare disease like this. So they use a lot of social media and the press to really advertise for their trial. And what they also said was that the retinal morphology was actually a better predictor of efficacy than age, right? That makes intuitive sense. So it's not so much that, younger patients versus older patients, but how much degeneration scarring has already occurred. And finally, they really stress the standardization of surgical techniques. This also makes intuitive sense because not everyone can do a perfect subretinal injection like L. McGuire can. So it's important to standardize this technique and be very, very specific about how these injections are done. Okay, so in summary, as you may have already gathered based on Barb and my talk, advancing from preclinical studies to clinical trials to regulatory approval to marketing is a really long and often confusing process. And most of us are certainly not experts in even a single step of this, not to mention all the steps of this. So, involving experts, and there are consultants who specifically help with various steps of these processes is really important. Definitely read these guidance documents if you're going through this process to help formulate trial design and marketing applications. A way to think about this is to start at the end point of where you'd like to arrive, right? Where do I want to go? What is the actual trial application I would like to what is the actual IND I would like to ideally submit? Or the marketing application I would like to submit, and work backwards to design the clinical trials to sort of arrive at that point. And finally, keeping lines of communication open with relevant regulatory agencies and doing this early is certainly a good idea. All right, and I'll conclude there. Thank you.
All right, thank you so much. Gus, you're up next.
Great presentation, Chi and Barb. Thank you everyone for this opportunity to share my thoughts here. I hope you can see my slides.
Yes, all looks good.
Great. So I'm going to be talking more on the design of a potential neuroprotection trial based on some experience and some of the literature out there today. These are my disclosures. What's relevant here is that I've been working with many of these companies when they interact with the FDA on their trials, and this is relevant to this presentation. So as we know, all the medications so-called to treat glaucoma are not really medications to treat glaucoma, but to lower intraocular pressure, which in turn which is accepted as a way to prevent progression. Although technically we don't have any medication in the market, at least in the US, the claim of which is to treat glaucoma or slow progression of glaucoma. And when it comes to interfering with the natural course disease, you can think about two potential ways. One is to we call neuroprotection, which is to stop or to slow the rate of progression of disease. Glaucoma is always progressive if untreated. So this is a strategy to target markers that could slow progression. And then you have the potential of improving visual function, which is something more recent. And the goal here is to perhaps reverse the damage that occurred and potentially even improve the quality of life of these patients. This concept of enhancement is not just theory. There's a number of studies that have shown that this is possible in the visual field. I just brought here one of the first ones that show that a significant eye pressure reduction can actually improve the sensitivity of the visual field, particularly in the points in the central part of the visual field. Our group, and Aki here is going to talk more about this, demonstrated that in a randomized trial, a small phase 2 trial, we were able to show some more improvement in test locations in the treated group versus placebo. And for those of you who are aware, this is very recent. On May 9 at FARVO, this company presented some results of went through an IND process of a novel treatment that also showed improvement in sensitivity and retinal fiber-layer thickness, and they're now moving to their pivotal trials. So how to design a trial? First, let's look at where things went wrong. So the memantine trial was the first attempt to do this through the FDA. And it was a large trial. We're talking about over 2,000 subjects, 4 years of follow-up, and actually 2 identical trials. But the problem is back then, not for the fault of the investigators, the technology and the knowledge was not as we have today. For example, visual fields were performed just one baseline and then every 6 months. They used FDT back then, which was how people detected an early functional loss. And instead of OCT, they had to rely on these photos. But still, there's some issues aside from that we need to understand. So one is that based on the FDA requirement at that time, they had to use the progression criteria that she just showed earlier. So at least 5 locations progressing significantly, either by the GPA or GCP or by 7dB, as she shows. So they went with the GCP, and that had to be confirmed. So we're talking about an event-based endpoint. It had to have 5 locations progressing. And when looking at a survival curve, your treatment group should be less likely to reach that endpoint than your placebo group. And the trial failed. All the endpoints tested, there was no significant effect of treatment aside from some trend towards those in a higher dose of memantine on the FDT, and after excluding the low tension glaucoma. So a lot of mining until you can see some effect here. So intent to treat analysis did not show anything. And some of the issues in terms of design that we have learned, one is with regards to inclusion criteria. One, back then they allowed patients who had, for example, filtering procedures done close to screening. And we know that those procedures slow the rate of progression significantly. So you're less likely to find endpoints in your population by doing that. They also allow patients with visual field MD worse than 10 dB, which is a range where the variability becomes very significant, and it becomes difficult to distinguish progression from just variability. Treatment, they allow the physicians at their discretion to change IOP-lowering treatments during trial. And of course, if a patient is progressing and the physician thinks there could be progression, they would enhance or escalate therapy, and again, affect the rate of progression in the trial. And finally, what I said earlier, visual field tests, just two a year is not enough, and they didn't have OCT back then. So what are the current FDA-approved endpoints? So I mentioned earlier, it's a long text, but basically what we're talking is about at least 5 locations of the visual field progressing, all right? Whether you're using a software such as the GPA today, GCP was back in the days, but the GPA today, or the cutoff of 7 dB that she showed earlier. And those have to be pre-selected in the case of the 7 dB endpoints. But more recently, after a number of companies reached out and pressure from the American Glaucoma Society, FARVO, American Academy of Ophthalmology, the FDA has shown some inclination to accept OCT as well. The problem is that, see how the definition of OCT here is very short compared to the other ones? And that's intentional because they don't have a specific criterion of OCT. Is it RNFL quadrants? Is it global RNFL? So we don't know, but they're willing to accept that, which is a victory. And what led us to do some of the studies I'm going to show you now is that whenever we came to the FDA and asked, okay, we want to try something different, the GPA or the 7 dB, such as how was done the memantine trial, you need a lot of patients and a lot of follow-up. Nobody has money to do like a 4-year study with 2,000 patients. So what can we do as an alternative endpoint? And the FDA nicely and collaboratively said, you need to show you that your alternative endpoints, whether structure or functional, predicts very well one of our accepted endpoints. So basically, if you demonstrate something alternative, whether it's in the visual field or in the OCT, that predicts one of those 2 endpoints that I showed you earlier, they would be willing to review that, and if that correlation is significant. So based on a lot of research from the past that showed that perhaps more important than an events-based endpoint of progression, such as GPA, perhaps looking at the speed of progression or rate of progression may be a better marker because that will allow you to see a numerical value, a continuous variable that you can show change over time. And also, based on the stage of the disease, you may tailor for each patient what is an acceptable rate of progression. Instead of saying patient progress yes or no, what does it mean? Progression in 2 years is a different scenario than a progression over 10 years. So looking at rates seems to be a logical way to move forward. And our group this is years ago, proposed that it was possible, based on what we had back then, to run a 12 to 18-month clinical trial as long as we use trained analysis, which I'm going to show you, and a clinically significant effect between treatment and placebo would be at 30%. Unfortunately, though, we didn't have data as we have today, and I'm going to show you, to demonstrate this, but that was out there, and people started to use that as basis for moving forward with their programs. Some of the pieces of information that became more evident over time were regarding to how to better use your visual fields. So we know that the 3 main factors that affect the variability of the visual field are the severity, the ability to detect progression, are the variability, which is associated with severity. More severe visual fields tend to be more variable. The frequency of testing, the more tests you do, you're more likely to find progression. And the actual speed of progression of the patient. If they're progressing more rapidly, you're more likely to detect progression soon. And this is the work from Chauhan and Ted Gary-Heath, showing different scenarios of rate of progression and number of tests done to detect progression. And another important study was by David Crabb and Ted Gary-Heath, in which they demonstrated the so-called wait-and-see approach, or clustering of tests, as we say today. So instead of doing like in the standard of care scenario, like in the left here, where you space tests every 3 or 4 or 6 months, evenly over time, you cluster the same number of tests, but now half of them in the beginning and half of them at the end, and recalculate the slope. And this was shown to reduce the variability and increase your power to detect progression. Putting now all the pieces together, as I said, number of tests, variability, and clustering, Felipe Medeiros compared through simulation models the number of patients that we would need to run a 2-year trial, comparing an even spaced paradigm, as we typically do in practice, the UK GTS, which was the clinical trial done in the UK for IOP lowering, that I'm going to talk later, that has some clustering. And then we have a real clustering design. We have 6 tests at the beginning, 6 tests at the end, and a couple of tests in between to allow to look for some safety signal, for example. And what they showed is that as you go through more cluster tests, you can come to a point that you're not talking about instead of 2,000 patients, but maybe a few hundred patients for your trial over 2 years. So we went from 2,000 patients over 4 years to now maybe 300 over 2 years by using this approach. And of course, it was based on simulations, computer simulations, based on real data, but you had to use real data to do the simulations. But the hint that this could be done in a short period of time came actually from the UK GTS trial, in which they used that clustering. And for those who are not familiar, this was a study comparing to IOP lowering with latanoprost versus placebo. And of course, latanoprost was better in preserving vision, visual field in this case. And what was interesting is that they showed a significant effect even at 2 years. But what was most interesting for me is that when they look at the data, even at 12 months, 1 year, there was a neuroprotective effect. Of course, this is an IOP-related neuroprotective effect, but it showed it's possible, at least with this clustering approach, to design a very short trial now. And now, again, based on the feedback from the FDA of what they wanted to see, so we did a study that we look at a very large database, and we defined these criteria here to include patients. So if you have your fast progressor, clustering of visual fields, and comparing of MD slopes, and to see it predicted the endpoints accepted by the FDA. So we showed that MD slope faster than 0.5 dB per year was highly predictive of reaching one of those 2 FDA endpoints, which they want to see, compared with patients who did not progress at that rate. So we brought that to the FDA, and they were actually very positive about it, with some caveats that I'm going to discuss later. But they basically said, okay, you did what we wanted to see. And it actually came also another study. Felipe did the same study. You can see that the curves here are remarkably similar between the two studies. And what was interesting from Felipe's work is that he also compared what would be the minimum difference in MD slopes between treatment and placebo that would be equivalent of reaching one of those FDA endpoints. And he showed that a 30% slower rate, as we had suggested in our previous data, will have the same effect as a 20% reduction in those FDA accepted endpoints. So gave a clue not only of how to use mean deviation, but what would be the expected difference between treatment and placebo. Of course, with OCT, things get even better in a sense of trial design. If you now combine OCT and visual field clustering and intense testing, we're now talking about even shorter trials, like the full trial with maybe 400 patients, 200 in each group. So there's some caveats, though, about using combined endpoints that we can discuss later, but at least show that it may help you, especially if you're like in the initial phase of your study. And more recently, Felipe looked at clustering of visual fields over 6 months, 6 tests in the beginning and 6 tests at the end over 6 months to see how it predicted progression over 2 years. And by doing that, as shown here, they showed that in 90% of the cases, the fast progressors over 6 months using clustering were the same fast progressors as 2 years in 90% of cases, which shows that potentially you can reduce your trial now to even 6 months, potentially. So in summary, how do I design my neuroprotection trial today? Well, the tripod for perimetry is what I said earlier. Using trend analysis, there are different ways. We're proposing the mean deviation, selection of fast progressors for inclusion in your trial, and clustering of tests at the beginning and at the end. Regarding OCT, it's a very viable and feasible solution. It's much easier to obtain than visual field. The problem is, my concern is that I don't think OCTs change fast enough that you can detect some change in a short period of time, whereas visual fields, you can see even improvement over a short period of time. And unfortunately, we still don't have a specific criteria of what the FDA wants. And finally, if you're not yet talking about a pivotal trial for FDA approval, but something more exploratory, there's some alternatives there. There's non-invasive methods to look at mitochondrial function that are encouraging. Electrophysiology is another one. But again, this is not something that the FDA is yet ready to accept, but it could use if you want to just find a signal for your study. Thank you.
Wonderful. Thank you very much. Our last presentation before we kind of open things up to discussion will be from Aki.
Oh, great. Thanks everyone for these wonderful presentations. Learned so much from all of you. Excited now to share our experience here. And, Gus is aware of many of these discussions since we're both at Columbia. And so feel free to chime in if you want to contribute anything else. So we'll be talking about real world considerations and challenges with ongoing studies in nicotinamide. I have no relevant disclosures. We'll be discussing the nicotinamide trials as a case example, talking about obtaining and maintaining the IND, as well as some challenges we've experienced in the nicotinamide trials and why having the regulatory support was really vital. So nicotinamide has been shown in numerous laboratory studies to prevent glaucoma in DBA2J mice, with higher doses being the most effective. 93% of eyes on higher doses did not develop glaucoma, which was considered a 10-fold decrease in risk as compared to untreated eyes, and 70% of eyes of mice on the lower dose did not develop glaucoma. So pretty good results in both levels of dosing. These encouraging animal studies inspired human trials, and there have been 2 pilot human studies. The first one was led by Gus at Columbia. And I'll give you some details of this trial. So this was a pilot study, a total of 32 eyes analyzed, 1 eye per participant. 21 individuals were randomized to the nicotinamide and pyruvate group, and then 11 individuals were randomized to the placebo group. All had bilateral open-angle glaucoma. All were adults aged 40 to 80 years of age. And the nicotinamide dosing was up to 3 grams per day, and pyruvate was also 3 grams per day, with a graduated increase in dosing over the first couple of weeks of the trial. The entire trial lasted about 8 weeks, so it was a relatively short-term trial looking at neuro recovery or neuro enhancement, and the primary outcome was improvement in visual function. So it was found that the group that was randomized to placebo had about 3 times more improving locations than the group that was randomized to placebo. This corresponds to about 25% of the entire visual field, so thought to be an impactful signal in the nicotinamide group. Another study was performed on nicotinamide use in humans, another phase 2 study that found improved inner retinal function using ERG when comparing to placebo. So these 2 phase 2 studies were found to be kind of complementary trials demonstrating neuro recovery with nicotinamide. And these studies prompted a larger trial in the US, as well as several ongoing trials going on around the world. And just as a reminder, nicotinamide is an over-the-counter supplement that's available in any health food store. And a little bit more about our trial. So we plan to have 188 subjects followed for a period of 20 months. And we'll have various outcome measures, some of which Gus sort of talked about in terms of perimetry and OCT. And we wanted to find a science-based vitamin therapy for our glaucoma patients. So when we submitted this to our institutional IRB at Columbia, we were surprised to see a determination from the IRB that an IND application would need to be submitted. The guidance from the FDA states that dietary supplements are actually not considered drugs and should not be subject to pre-market approval requirements unless the clinical investigation evaluates the dietary supplement's ability to diagnose, cure, mitigate, treat, or prevent a disease. And this determination set the course toward obtaining an IND and partnering with the FDA and obtaining support from our institutional clinical trials office. So I'll talk to you a bit more about that now. The FDA's role begins once the drug's preclinical testing has been completed and the drug's diagnostic and therapeutic potential in humans is now to be tested. As all of the prior speakers mentioned, it is great to be involved with the FDA as early as possible. And the Center for Drug Evaluation and Research, this is a division of the FDA that works specifically on drug approvals, has a pre-investigational new drug application IND consultation program. So if you start talking to them early on, you'll have an idea of exactly what they are looking for. There are several types of INDs. So we filed the individual investigator IND. This is submitted by a physician who initiates and conducts the investigation. It can be for an unapproved product or an approved product for a new indication. Of course, for an over-the-counter supplement like nicotinamide, it never underwent approval, but we were saying that we were applying it to glaucoma patients, and that was really why we needed the IND. There are also multi-center INDs. In this, our study has eventually actually became multi-center. Stanford is also now a center that's recruiting for our trial. We didn't have to do too much more to get them involved, aside from informing the FDA. But if you initiate a new multi-center trial and you plan to have a central IRB that conducts reviews on behalf of all the study sites, the process is a little bit different, and you can continue to work with the institutional IRB to some extent, but the central IRB will really be spearheading the whole operation. Importantly, if you're doing a multi-center trial, the IND will also allow the drug to be shipped across state lines. Otherwise, the drug needs to be approved in order to be transported legally. There are also emergency use INDs. In these cases, actually, there is an experimental drug in an emergency situation that doesn't allow time for submission of an IND. This is also used if the drug does not meet criteria for an existing study protocol. And then there are treatment INDs for serious or immediately life-threatening conditions. The distinction between this and the emergency use IND is that an IND is actually submitted for the treatment INDs. IND application, there's so much to talk about here. This is just a very brief summary of what it involves. A cover letter, specific FDA forms that are available online, and then as much data as you can get your hands on the product. And this includes clinical, preclinical safety data, the labeling that the product is going to have, drug formulation, composition, quality control, storage data, and so forth. All the preclinical data that you can provide, animal study results, rationale for human trials, and then the clinical information on how you plan to test this product, the clinical protocol, everything from your IRB, investigator qualifications, what you plan to use for informed consent. And then, there's a little bit of, should the IRB go first or should the IND go first? Usually the IRB is not going to approve the protocol until you have the IND. So you can go back and add in the IRB approval once you have it. And then if there's any previous human experience on the drug, you can add that in as well, and an assessment of the environment. Here are just screenshots of these FDA forms. They're actually very straightforward. Most of it is just kind of, demographic and kind of office information. The protocol itself is what ends up being the longest part of the IND, but you'll need that anyways for your IRB, so it's really not too much more work. And if you can take anything away from my presentation today, I would say try to lean on your institutional's clinical trials office as much as possible. Ours was incredibly helpful to us throughout this whole process and really helped us in reviewing all of the documents and making sure that we were meeting all the regulatory requirements. So they are the ones who, once we received that determination from the IRB that we probably need to get an IND, they're the ones who said, yes, indeed, you do need to get an IND. They said they always recommended multi-center studies to be submitted to the FDA, like I said, because the drug's going to be shipped across state lines. And, if your product is already lawfully marketed in the US and it is listed as an approved drug on the FDA's National Drug Code, then you can complete this worksheet that they provided to us that helped us determine whether or not we actually needed the IND. And of course, your product would need to meet all of the criteria listed here in order to be exempted from the IND. A little bit about the role of the IRB. Of course, we've all used IRBs for numerous other sorts of studies, but in a situation where an investigational new drug is being evaluated, the IRB is going to regularly audit your work and, of course, investigate any incidents that it deems might be associated with non-compliance of the FDA rules. The IRB will collaborate with the sponsor investigator, the IRB chairs, and the clinical trials office to create a corrective action plan if it seems like there are cases of non-compliance. And very helpful, they'll send us email reminders all the time about various FDA filing requirements. Our CTO has helped us so much in meeting with us, preparing documentation, reviewing all the documentation prior to submission. We consulted them pretty early on once we had heard back from the IRB, but actually late in a way because we had already developed our whole trial before we reached out to them. They also have all sorts of training for coordinators who are going to be very involved in this process, of course, as well as the PIs. And our clinical trials office required that we meet with them in person prior to initiating this process. And, they've helped us with our annual reports to the FDA, as well as to our DSMB and our internal monitors. And they've helped us retain all the necessary documents and so forth. FDA inspections, we have not yet had one, but we always hear about them happening. So if a PI is notified of a scheduled FDA inspection, the PI is supposed to go immediately notify the IRB, the clinical trials office, the office of the general counsel. And the CTO and the IRB will review all the inspection procedures and really will assist you through the entire process. So I then now wanted to just touch on an incident that we had in our trial, and which helped me realize how important the regulatory support was, because I got to see how helpful they were. So this is a study this is a case report that we published in Journal of Glaucoma. This was a patient enrolled in our nicotinamide study, 73-year-old woman with POAG, not a very significant past medical history, just endometrial cancer, now in remission, osteoporosis. She was on a statin, low-dose statin daily. She had initiated the study drugs and increased her dose in a graduated fashion per our protocol. We had asked participants to use 1 milligram of study drug A and study drug B for 1 week, and then escalate to 1 gram for 1 week, and then 2 grams for the next week, and 3 grams for the following week, and then to be maintained on that target dose. After she was on the target dose for 3 weeks, she reported vomiting and was advised to stop all study-related drugs and seek medical attention immediately. Her lab work revealed an elevated liver function test. AST and ALT values were very elevated. ALK phos was also elevated, and she had leukocytosis. She was admitted to the hospital, treated with intravenous N-acetylcysteine, and she was found to have presumed drug-induced liver injury with coagulopathy. There was no secondary cause of hepatitis that could be identified, not infectious, autoimmune, or any other cause. And so at this point in the study, we had to ask ourselves what we should do. Of course, we were worried about the patient's health. We were happy to see that she was in the hospital and getting the treatment that she needed, but we were very concerned about her and very concerned about the other participants. So we had a few different options of, either continuing the study and informing some of the regulatory bodies, informing the participants, or continuing informing the regulatory bodies, not informing the other participants, or stopping the study, putting it on pause, and investigating further. We chose the last option. We had a voluntary study pause. We informed the DSMB, the FDA, the IRB. Our DSMB and our investigators met with hepatologists. We also added a hepatologist to our DSMB. We analyzed the study pills to make sure that our participants were actually getting what we had asked them to use. And we ended up modifying our whole protocol to include LFTs at baseline and throughout the study, as well as stopping rules based on LFT elevation. Thankfully, our patient's laboratory work normalized, and she was discharged from the hospital within 3 days of admission. She was transitioned to outpatient hepatology, and then now she only sees her internist and does not have any long-term effects from this adverse event. But it could have led to all sorts of, kind of terrible things for her. So this was all on our mind. This event occurred in August 2023. And after this whole process, the IRB and FDA allowed us to restart our study a couple months later in November 2023. A similar case actually has occurred in Singapore in one of their trials. 69-year-old woman who increased her study dose, was on 3 grams per day for 10 days, kind of similar duration to our patient, and she was found to appear icteric and had some other symptoms at her clinic visit, found to have elevated AST and ALT as well. She discontinued her study products and her labs normalized. In this case, the person had a very low BMI of 15. That was not the case in our study in the US, but another person with a similar situation. There has not been a whole lot published on nicotinamide and its safety. There are many other studies on nicotinamide that have found some toxicity at very high doses, but the dose that we were using, 3 grams per day, was thought to be relatively safe based on a variety of studies included in this review paper in the diabetes literature. Interestingly, it is 150 times the daily recommended dose of nicotinamide, which is just 20 milligrams per day, but is based on some of the laboratory work on mice, showing that the low dose, which was efficacious in 70% of my mice eyes, was 2.7 grams per day for a 60 kilogram human, which is pretty close to the 3 grams per day that we have in our study. Niacin, which is a different form of vitamin B3, has been associated with liver injury, but nicotinamide hasn't prior to these reports from our studies. And nicotinamide at very high doses, like I said, up to 9 grams per day, is associated with hepatitis, but these 3 grams per day prior to our findings was considered acceptable. Of course, we know that a large proportion of the US population uses supplements, and patients often inquire about using nutritional supplements as complementary or alternative medicine. It's possible that many people are using nicotinamide at high doses without physician oversight, maybe reading from some of the research papers or other popular literature. And patients may not have reported it, physicians may not have suspected it, and the whole industry is sort of unregulated. So based on that, we ended up creating a position statement on the use of nicotinamide for glaucoma neuroprotection that really includes many of the ideas that we talked about, limiting doses of 3 grams per day to just the clinical trials. And if one were to use a smaller dose of nicotinamide outside of clinical trials, to be monitored very closely with serial LFT testing and so forth. So in summary, there are many reasons why an IND might be required, even for an over-the-counter drug or supplement. Important to engage with the FDA early to get their advice on how to best work with them. Your institutional clinical trials office is really a fantastic resource. They held our hands every step of the way. And importantly, the IND and the regulatory bodies will provide you with the legal and ethical support for your study, especially if you encounter unexpected situations. Thanks very much for allowing me to speak, and I'll turn it back to the group.
Thanks. I really appreciate all the varied perspectives and kind of firsthand experiences with a lot of this. I want to open things up to everyone in the group. If there are questions, comments, items of discussion that you want to bring up, now would be a good time to do it. I see one question that was in the chat, and Barb, you answered that in the chat, but I think it would be good to answer that out loud as well. And the question is, from Brent Young, when evaluating safety of a cell transplant therapy, do you need to prove you looked everywhere in the body for possible cell migration, or are there particular regions that are more likely for accumulating cells?
And what my response was it really depends upon how and where you're delivering it. So the requirement is obviously much lower if it's contained within the eye, versus if you were giving something, IV or into a local organ. So it really comes down to what's the likelihood that, a stem cell would leave the eye, right? And again, it comes back to, what's the mechanism or what would be the safety concern? And it would probably be unlikely, but again, I'm just kind of making this up as we go. Could you imagine that a stem cell could be secreting something that could have an impact elsewhere outside the eye? And that would be the way that, people would think about it.
Okay, great. Other questions? Felix Trachtenberg, I assume that you had a question there. It looks like a 5-part question. Would you be willing to unmute yourself and ask those questions to the group over Zoom?
Yes, of course. So I don't know how many questions I can ask given the time constraints, so please stop me.
Sure.
But first question I want to ask, in terms of the species, so FDA prefers 2 species, right? And in terms of NHP being required, if it is required, do we still need rabbits after rodents? In the past, rabbits was the preferred species, but now if requirements are, shifting towards the NHP, are the cost justifiable, etc., because it would be the 3rd species because we started with rodents. And then, in terms of safety versus efficacy, obviously safety first, so you need safety in all the species. But in terms of efficacy, do we need to aim for and invest in efficacy testings in all the selected species, etc.? I mean, just to get through the door. So that's question 1 and 2. And I can finish other questions, or if someone wants to respond. And if
Yeah, maybe let's get a response to question 1, and then we can keep moving on to question 2. Anyone have any thoughts?
Gary Novak, can I try that?
Yeah, absolutely.
I'm going to talk about safety, not efficacy, and Barb and others can join. ICH guidelines are 2 species, and you need for ocular, as Barb said, depends on route. If you're giving it by the eye, probably need 2 species ocular GLP tox. Probably also need at least 1 species systemic. That's if you have homology. If your target is not in a lower order species like some of the VEGF. Sorry if this is too much detail. So the VEGF that's in I'm going to use in pagatinib and ranibizumab is only in primates. So that's then there you can use primates only for your tox. Does that answer your question?
Okay. All right. Thank you.
And I could just chime in a little bit. So for Cularis, we're studying a small molecule, and the most sensitive species was dogs. So we never did any of our studies in NHPs. But all our systemic work, to Gary's point, even with a topically administered compound, you always worry about systemic exposure. So again, we gave high doses systemically to dogs to elicit any risk of a safety concern.
But wasn't it you or someone else who mentioned that nowadays for glaucoma, FDA requires NHP?
I don't.
So that was not me.
Clarify.
Oh, it was.
Sorry.
I thought someone had it on their slide required next to non-human primates, or I may have overlooked.
I don't know the slide. The guidance, ICH guidance is 2 species, 1 rodent, 1 non-rodent, and rabbits are our rodents. And the higher order species, to Barb's point, can be dogs, mini pigs, non-human primates. Sorry to be terse, but I'm trying
Isn't shrews, aren't they looking at tree shrews too? I think,
for toxicology or
I
for
don't know.
pharmacology?
Maybe efficacy. I know that's come up a few times.
Armadillos, big, no, just kidding. There's a much bigger issues. You want normative data in that species so that when on Barb's brand new compound, they say, Barb, we found that in 5 animals, it turns out they're really hungry for almonds. And we've never seen this before because we've never used tree shoes. So we don't know. How'd I do, Barb? Is that?
That's a great point, because that actually was, you remember the company you helped me with a few years ago.
Okay.
And we actually.
Did it start with an A? Okay,
Yes, started with an A. We looked at a compound, topical small molecule, and we worked with a rabbit species, Dutch-belted rabbits, that there's not, to Gary's point, a lot of normative database, and something showed up on safety. So we then had to prove that actually the safety signal we were seeing was not related to the drug. So you have to really think carefully about what species you're looking for and what you're asking it to do.
So I can answer that question. I did have that word on one of the slides because one of the guidance documents did mention something about non-human primates being strongly recommended. But, Gary and Barb, you guys are the experts. So certainly it's not a requirement of the FDA, but it was something that was strongly recommended. So I had that in parentheses and not as a definite.
Yeah, that's what you did have.
Right, thank you, Kate.
Yeah, I saw that.
May I have a chance for another question, or someone else, I think, already is typing a question?
Sure, go ahead, and then I think
I'll skip them to the question 4 that I typed regarding considerations for co-treating with some drugs that reduces intraocular pressure in a case when your drug is actually gene therapy that is neurotherapeutic to protect and perhaps regenerate RGC soma and axons. So in a clinical setting, you probably would not want to deny a patient a drug that reduces an intraocular pressure if they have progressive type of glaucoma. If you want to also test, in addition to that, your neuroprotective gene therapy. So in terms of this consideration, when you study your specific product, gene therapy, how do you go about this consideration? Do you have to do it first in human trial, for example, together with yeah, with drugs that reduces intraocular pressure or not, and if there will ever be a need for a group without intraocular pressure reducing drug for this type of glaucoma, and so what would this imply for preclinical animal studies? Would they also be all need to be conducted together with such intraocular pressure reducing drug? Because typically in glaucoma mouse glaucoma model, you don't do it. You just do your neurotherapeutic, and doing it together is not a standard way. But for IND, etc., if it's a requirement, that I'm just trying to seek feedback on the strategy, how to plan such studies.
I think that's
I
a
can,
Gus question.
yeah, I think I can start. So I don't think there's room for no IOP lowering treatment at this point. All the interactions I had, you need to offer standard of care to these patients. What you can do, though, and people are looking at that, is contrary to the Memantine trial, establish clear rules about treatment in terms of escalation during your trial. For example, in a shorter trial, you could consider not allow escalation, assume that your population is not that severe, and because imagine like 1 dB per year, which is a fast rate of progression, it translates to a small amount even in 2 years. So you could consider that, although you should still have a rule for escalation when needed, but not to have a fully non-IOP placebo group at all, from my understanding.
Question I'm seeing in the chat directed to Aki. Thank you for the informative talk. This is from Sung-Soo Ro. So was the IND application mandatory for your current nicotinamide trial of 188 participants, or was it just for the legal protection against the unexpected bumps?
Good question. So this IND was mandatory for this larger trial. So we did a similar trial, short term, same doses of nicotinamide and pyruvate at the same institution in 2019. And that trial did not require an IND. The IRB did not recommend that at the time. But for this larger trial, they did recommend an IND. And I think it may have been like a semantics thing or something along the lines of something written in the protocol made it seem like it was more important to have an IND. And the specific wording is that, nicotinamide and pyruvate were being tested to see if they could help prevent glaucoma progression, just wording you'd use for neuroprotection, essentially. Whereas the initial trial, the pilot study, probably had different wording talking about nicotinamide and how it may enhance visual field test taking ability. And so specific wording to diagnose, cure, treat a condition, which is how it's interpreted in the larger trial, led to the IND.
Yeah, just to add to that, you're absolutely right. So for such a short period as the one that we did the pilot study, it was not assumed that we'll have any treatment of any kind. But we're also, the main study question was actually the performance of visual fields, which could have been, for example, and still could be, that these patients get more attentive and have a better performance during the test. So that was more like a behavior than actually a treatment for disease.
And
Go ahead.
Matt, can I just sort of chime in? Because something that Aki said was really, really very important is that this was a supplement that was a dietary supplement. So it had never been approved for an indication. But the other thing you mentioned, Aki, too, is that if you're taking a drug that has gone through an IND application and now using it for a new indication in the eye, you can reference that prior IND and or the NDA. So you can always reference the work that the FDA has already seen to get a compound drug to a certain point, even if the drug was never even approved. So if the IND was open, the FDA has seen it, you can always reference that. So that's helpful. I don't know if Gary has any thoughts. This is his area.
Lots of what ifs. I think I would say it if it's an approved drug off patent, it's easier to access. Approved drug on patent, a little more questionable. Unapproved drug, I don't think so.
So I have, sorry, go ahead.
Matt, do you want to go first, or should I ask a question first? It's up to you.
Sure, I'll ask real quickly. So, Restore Consortium interested in, replacing ganglion cells, regrowing optic nerves, that kind of thing, and so it's sort of applicable to a wide variety of optic neuropathies. And, so there's a question sort of related to animal models for efficacy. And, I recognize and I learned today that, kind of the primary thing at the early stage is safety with sort of efficacy as a little bit of a secondary. But if you're modeling a disease in an animal, even if it's sort of you're getting up to the point of a non-human primate, um, how close does that have to be to the human condition to sort of qualify for having demonstrated some level of efficacy in an animal study, in order to be considered valid? And
Can I try that again?
absolutely.
I'm just an unofficial observer here. So Marco Zarbin, who many of you know, asked me that question whenever ARVO was first in Seattle. Was that 10 years ago? And it really applied to inherited retinal disease. And the answer is, it seems counterintuitive, but for regulatory purposes, for a drug, for CDER, you don't have to cure the disease in animals to try it in people. CBER is a little different. And then to Key's point, sorry, I've spoken at a symposium and I continue to say your name wrong. I'm sorry. But, Luxturna it's gene therapy is very different. And the advantage there, as you pointed out, is the Briard dog that had the same gene defect as humans. So you may, for science reasons, you may want to show that, and it may be more important for gene therapy. For regulatory reasons on CDER, you don't have to prove it cures the disease. You should show that you get the drug where it needs to go and it has some activity. But that's different than curing the disease. Your investors may feel otherwise.
Got it. Okay, thank you. I think I kind of cut you off earlier.
No worries. I did want to give a shout out to Gary. Thanks for joining us, Gary. He gave a very nice talk at a symposium we did recently, also with RReSTORe and with Tom as well, just sort of examining basically this question at ARVO. And he talked about the IND process. So he's very much an expert. So if anyone has questions, definitely reach out to him. So my question is for Gus. Gus, you have presented some really nice data. A lot of it you've done yourself, just looking at how to sort of decrease that, beginning part of starting clinical trial to when we can actually see whether there's an effect. So that's great work. You presented that paper, I think it's done by Felipe, where they looked at the various effects of clustering at the end. And I guess my question with that is, it looks like he showed 6 clusters in a visual field test in the beginning and then a 6th cluster at the end. Is there an optimal number of clusters of visual fields? Is 3 sufficient compared to 6? Is 4 sufficient compared to 6? Would 9 be better? Just try to get a sense on sort of the practicalities of sort of clustering.
Yeah. And that's where your trial will have some issues, is how to do that with the patients.
Right.
They don't like to do those tests, and
exactly.
takes time. So we did some statistical modeling on that, and from our own work, it seems that after the more tests, of course, there's always going to be an incremental effect, but it kind of plateaus after 4. So even Felipe, talking to him last time, he thinks that, or he actually showed that if you did not use all 6 but 4, you had like a similar effect. So and that's more doable, I think. And the other thing about that clustering is that although the model suggests that you're doing everything in the same day, it's not. You can actually allow, let's say, that the 4 tests are done within a week or 2, for example. As long as relative to the entire duration of your trial, that becomes a small period of time. So I say 4.
And are these tests like 24-2 CETA standard? Is there an effect of, doing a, CETA fast or 30-2 or something like that?
Yeah, so a lot of this work was based on modeling using CETA standard data. And his actual trial that I also showed also used CETA standard. And it is also, of course, it's going to be easier for the patient to do CETA faster. The knowledge about variability, though, is not as well known as we know for CETA standard, especially in areas of lower sensitivity. The studies that Andres Hale published show a very comparable test-retest variability for CETA fast, CETA standard, CETA faster. But if you look at the graphs, once you reach thresholds of abnormality, they actually have a much, not much, but have some higher variability. And I think, especially in a trial that you want to do for change in a scotoma, whether improvement or worsening, that could be problematic. But I don't see why you could not also try that.
Thank you.
Barbara, see your hand up.
Just a quick question, Gus, because I sort of had the same questions as Key. 24-2 was what you were showing. What about a 10-2? Could there be, because again, 10-2 can pick up some defects earlier. I mean, do you think it would have a better likelihood of showing small changes in sensitivity?
Yeah, so we actually have some work using a clinical study that we're doing all those tests on the same day to look at test-retest variability, and the 10-2 indeed has less variability, even if you just compare it to the central points at 24-2. Could be because of some more averaging, 68 averaging versus 12, but it's there. And there's a paper from Bob Weinreib's group that showed that you can detect change sooner on a 10-2 versus the central points at 24-2. What I think is a much more interesting question is, what is a fast progression for the 24-2 may not be what a fast progression is for the 10-2. So that has to be taken into account. But Aki, do you want to say something about the ongoing trial and what you guys are doing?
Yeah, sure. So we are, based on Gus's advice and other folks as well, we are actually following our patients with 10-2 cluster testing. They are getting 24-2 tests for safety reasons. Obviously, we want to monitor their peripheral field as well, but 10-2 will be one of our study endpoints.
Oh, very cool.
For many of the reasons that Gus mentioned.
Makes sense.
All right. We are at the end of our scheduled time together, so I want to thank our facilitators for their presentations and sharing their expertise with us. What I'm going to do is I will email the email list where I sent out announcements for this event once we have the video of this posted up on the RReSTORe website. And then always we're looking and eager for your feedback. So if you have comments on things we can do to make these events better, feel free to email them directly to me. Or if you have topics that you would like to bring up in future discussions, things that you think would be useful for the community that we could try to find some expertise on and bring up for discussion, that would be really helpful as well. So feel free to send those ideas to me. My email address is matt.vanhook@unmc.edu, and you can just find it in the announcements that I've sent out for this event. So again, thank you. Thank you all for your time. Thank you for a good discussion. I hope you all have a wonderful afternoon.
Thanks, Matt.
Everyone, thanks, Matt.
Thank you.
Thanks to you. Yes.
Thanks, Gus.